I'm your host, Richard Johnson, and you're watching The Late Show. This week's topic on the show is genetic engineering. Are we ready for it? Are we not? I mean, I know I'm ready for a robot arm, but is society ready? We have a series of 5 specialists in each aspect of genetic engineering, and they are going to tell us their opinions on whether or not we, as humans, are prepared to genetically modify ourselves or not. So, Dr. Walsh, you're a leading scientist in the lab organ growth, and you're the proponent of these type of projects, correct?
Yes, Richard, and there is a reason for that. I believe that lab-grown organs are not at a point where we can take this technology, say, on a scale that is nationwide. There's still a lot of things to work out to fix, but I feel that once we get to this point, we should absolutely go forward with starting to grow organs for people to try to save people in any medical field, in our profession. If we can do a single thing to prolong someone's life, I feel that we should do that. We should not be waiting 10 or 15 years to try to get some sort of approval from places like the FDA before we initiate these products, because in that 10 or 15 years, you're losing so much life. So that's just how I feel about it.
So I understand that there was a big break some years ago in your field. Can you tell me more about that?
Yes, there was. Several years ago in the early 80s, I believe it was, a man by the name of Dr. Joseph Vacanti was actually on vacation of all things, and was noticing some seaweed move in the water. It's a long story, but he realized that if we used biodegradable polymer, we could grow thicker tissues, and then as the tissue grew, this polymer that it was growing around would dissipate, would go away. And prior to this, we could only grow just thin pieces, less than the thickness of a dime, of tissue before it died from lack of oxygen. And so, with that breakthrough, we were almost immediately able to start growing things like ears for people that had been involved in fires and that sort of thing. And so, with this technology, as soon as it becomes available, we need to implement it. I don't know what the big deal is, what the difference is between something on the outside of a body, because the ears did not bring much protest. We were growing facial features and stuff for people, and that was not a big protest. But for some reason, when we start doing stuff inside the body, it is. And so that's just how I feel about it. We need to start implementing this stuff almost immediately when we have the technology to do so.
Good evening, kind sir. I am honored to be on this show tonight to be talking to you about the fault in this new trend of designer babies. Well, you see, these types of altercations can cause serious damages to the embryonic development and the fetus itself. The argued advantages of the new neogenetics is the reducing of risk and disease and suffering in children. Yes, I do understand that look on it, but it's not that simple. A trial had been run by a gathering of young adolescents and strategic learning on this look into this process of designer babies. The trial was established after the consideration of diseases linked to XX chromosomes. Once the experimentation had been examined to full extent, the conclusion had come to that the only need for this type of gene altering would need a verified special ill or disease case. The issue that arises why people would actually be performing these altercations on their children can really vary. One gender could begin to dominate another, or overpopulation of just the human race. Another thing to take into account is the emotional and psychological changes that can really alter in a gene of a child. All in all, I think we should just stay away from designing babies.
Welcome back to The Late Show. I'm your host, Richard Johnson. Joining me today is Dr. Justin Martinez. He is a specialist in genetic engineering, specifically the cloning aspect. So, Dr. Martinez, cloning humans, should we do it, should we not?
I'm glad you asked. Advances in genetics, science, and technology allow us to the possibility of being able to clone human beings. However, whether that technology will be safe and effective as well as legally permitted is pretty uncertain. I don't think we're ready. It will undermine our sense of self and identity for 3 reasons. First, cloning would undermine our sense of individuality and uniqueness. But it could only undermine our genetic uniqueness, not our full individuality. Secondly, cloning would undermine the value and the worth of human beings, but it would not make individuals replaceable or of any less moral worth. And third, a clone's freedom of autonomy to construct his or her own life would be undermined by the presence of an earlier twin, of an exact replica.
Right.
But only a mistaken belief in genetic determinism supports this feared loss of freedom.
So, Dr. Martinez, let me ask you, would you clone yourself?
I don't think I would. I would be too afraid of my clone getting us into trouble,
Right.
And also, I don't think people would take my clone seriously. They would just disregard it as a science experiment.
Richard, thanks for having me on the show tonight.
Of course.
I'm excited to be able to explain as to why I believe, but not only me, some other scientists and doctors believe that genetic engineering can be a useful technology on humans for this generation and generations to come.
Well, that's why you're here tonight. Many people wonder about genetic engineering and if it can be done on humans or not. What are some ideas that make people worry if it can be done on humans or not?
Well, we believe that as long as the correct steps are taken, that indeed genetic engineering can be a useful technology on humans. There are several typical arguments that are brought up within debates about genetic engineering. Some of those examples would be the overregulation of genetic engineering, the opposition view of binary polarization, and just the negative consensus overall as genetic engineering as a whole.
With my very limited knowledge about genetic engineering, one of the main things I know that holds it back is the overregulation on the topic. Can you tell me more about that?
Yes, overregulation is a big argument that is continuously brought up when speaking about genetic engineering as a whole. Back in 1989, the National Research Council did a field test on genetically modified organisms, and they found that they would be able to predict the physical expressions of the genes. Now, with the limits and regulations that are imposed on genetic engineering, this implies that the hypothesis shouldn't be tested, whereas this brings out the anti-scientific theories of the argument and it exposes them. Scientific hypotheses are meant to be tested, not excuses not to be tested.
Hello, Richard. Thanks for having me on the show.
Of course.
So you know that I've been a pretty adamant opponent of genetic engineering with CRISPR and Cas9 gene editing tools.
Yes, as I understand it, CRISPR allows us to take a segment of a person's DNA and replace it. Why are you so adamant against its use on people?
So yes, CRISPR actually refers to the segment of DNA that informs the Cas9 enzyme of what to modify, and I'm adamantly against it because it's an unpredictable process in nature. In 2015, a team of Chinese researchers were able to modify the DNA of triponuclear zygotes' DNA, but what they found was that the process left them mosaic. So the zygotes did not have all of their cells' DNA modified.
Correct. So you're saying that the process isn't ready today, but it possibly will be in the future?
Maybe, but the technology still has a long way to go.
Correct.
Oftentimes the process won't even change the DNA properly. It'll, modify it, but won't do it exactly as the instructions want it to, which can lead to maybe innocuous problems or potentially life-threatening issues. So I'm not saying we shouldn't research the technology, I'm just saying that it should not be used clinically on humans at the state it is now.
Well, thank you for coming on the show.
Yeah.
Today's guest is Professor Kayla Garcia, who is part of the genetic research team at one of the top universities in the nation, who is currently working on stem cells. So tell us, Kayla, what sort of potential do stem cells have?
That's a funny question you asked, Richard, but I'll give you the answer. The life-saving potential locked within stem cells is limitless. Stem cells are undifferentiated biological cells that have the capability to take form of any type of cell in the body while multiplying and renewing itself. This means that doctors could place these cells in an uninjured part of a body, and they will proceed to help and regenerate alongside other cells in that body as long as the person is still alive.
Indeed, that does sound limitless, but what's the big holdup? Why aren't we sticking these suckers into people like tonight?
Well, none of our trials have been completely successful, although we have had partial success. We know for a fact that stem cells have regenerating capabilities, but they do not have the longevity to be effective in a body. We see them work, but we do not see them stay. They die off eventually, therefore we can't risk to have such an unstable cell inside a body.